Hello, my name is Matt. I'm a final year PhD student at Birkbeck College in UCL. And I'm going to be telling you a bit today about the work that I've been doing on computational analysis of protein structure using chemical cross-linking mass spectrometry data. Proteins are three-dimensional molecules that perform many essential functions in living cells, and understanding their 3D structure is important for learning how they work. So in the Topf lab, headed by my supervisor Maya Topf, we work to produce algorithms that model protein structures using different types of experimental data. So this data can be from cryo-electron microscopy, X-ray crystallography, or various others. One type of data that we're particularly interested in is mass spectrometry, which is the technique which my second supervisor, Kostas Thalassinos, is interested in. So my research is aimed at producing computational methods to model protein structures using data from a technique called chemical crosslinking mass spectrometry. So proteins are made up of chains of things called amino acids, which we can refer to as residues. And these residues fold into different conformations which form big 3D structures. So with chemical crosslinking, what we can investigate is which residues are, first of all, on the protein surface, and two, which residues are close together in space. And this is what we see from these things in red called crosslinks. These are from when we react a protein with a chemical crosslinking reagent. And if they react with residues, we can see, first of all, that they're on the protein surface, and two, that the residues linked together are close together in space. So this is important data which is used by quite a lot of people in the field, and we have previously devised computational resources to score model protein structures with this information. A lot of my project has been focusing on another type of data we get from a crosslinking experiment called monolinks, which are shown here in green. Now, these are when a crosslinker only reacts on one end with one residue in a protein structure. So they don't actually give us any distance information. They don't tell us which parts of a protein structure are close to each other. But what they do still tell us is which parts of a protein are on the protein surface. And because we get more of these monolinks in an experiment than we do crosslinks, we have more information to actually model protein structures on. And we recently published a paper in Structure where we first of all designed a scoring function for scoring model structures based on this residue exposure information. This is called the monolink depth score, where we score model structures based on how deep these residues, which have monolinks attached, are in this model structure. And then we can show that when we combine this score with our previously defined score for scoring crosslinks called the matched and non-accessible crosslink score, or MNXL, we can see an increase in modeling performance. So we can model protein structures better when we combine these two types of information. This is something that the field is currently not doing, so we are really trying to show the usefulness of this data, which we already get from an experiment, but is currently not being used to model protein structures. So many different people have helped with this work. So I want to thank everybody in the Topf lab, everybody in the Thalassinos lab, my thesis chair, Mark Williams, Dave Holdershaw for his IT expertise, MS Madhusudan, who's our collaborator in India, who's also worked on our paper, and Birkbeck for funding our research. And I hope you found this an interesting insight into my research, and I thank you very much for listening.